So welcome to today's webinar on bioinformatics for precision medicine. We know that precision medicine is actually changing the way we understand, diagnose, and treat many diseases, right? That also include cancer. And this transformation of the way that we treat diseases is actually driven by high-throughput sequencing data, that is high-throughput sequencing molecular data that we get from patients, for diagnosis and prognosis purposes, and for animal models, to actually understand the disease that we are actually dealing with. And we can also perform large-scale cell line experiments to characterize the responses of different diseases, and specifically tumor also, to several of the treatment options that we have lined up for them. So to compare with precision medicine, traditional medicine uses symptoms to diagnose diseases and drugs to treat these symptoms. But precision medicine aims to turn this approach on its head. Right? Precision medicine is an emerging approach for disease treatment and prevention that takes into account individual variability in genes and individual variability in environment and different changes in the lifestyle for each and every person. This approach, or such an approach, will allow doctors and researchers to predict more accurately which treatment and what kind of prevention strategies will work for a particular disease, and in which groups of people it is expected or it is understood to work better. So this is in complete contrast to one-size-fits-all approach, that is in which the disease treatment and prevention strategies are developed for an average person with less and less consideration for the individual variances and individual differences. So after a lot of research, we are poised to enter a new era in the medical practice where detailed genetic and other molecular information about the patient's disease is routinely used to deploy effective and patient-specific remedies to treat it. We are about to enter the era of precision medicine. So the future of medicine is increasingly driven by the availability of data and the expansion and the standardization of high-throughput sequencing that results in medical practice that is both ultra personalized and ultra precise. Extracting meaningful implications from this data requires deep analytical expertise, and it also requires integrating several of these data or several of these omics data from different molecules. So let's take a look at what we got here and what drives this change in the modern medicine. Actually, the role of bioinformatics in general in the context of diseases is to allow researchers to access, to process, and to analyze and also to visualize several of these kinds of high throughput sequencing data that we will get introduced to over the course of this program. And this program will also introduce you to several examples of how this connection between this omics data and their explanation or the reason behind the diseases and their effectiveness into several treatment options was done successfully by researchers studying, for example, viral infections like Ebola virus outbreak in West Africa. We will also take examples from COVID-19 pandemic, as well as we can also study microbial pathogens causing, for example, tuberculosis, and development of resistance to several of the treatment options and antibiotic that this tuberculosis encounters it to. We can also consider cancer and their responses to different treatment therapeutic options that we have. And in the end, we will also have options to work with single cell parasite that causes malaria. So we will briefly touch on viral genomics, viral evolution, and gene expression patterns and detection of significant genomic variants, as well as some important aspects of microbiome and virome community in a host that are being studied to find important associations of these several different omics data with disease susceptibility and disease outcomes as we move along. Yeah, my animation is running slow. So cancer, I don't have to introduce all of you to cancer. Cancer is the leading cause of death worldwide, and this accounts for nearly 1 in 6 deaths, right? Some of the most common cancers, as we know, are breast cancer, lung cancer, colon cancer, rectum cancer, and prostate cancer. Cancer-causing infections are prevalent, and they are such as papilloma virus and hepatitis virus are responsible for approximately 30% of the cancer cases in countries that are still underdeveloping or still developing. Right? So many of these cancers can be cured if they are detected early and if they are treated effectively. And this is where precision medicine is applied to enable early detection of cancer by studying various different molecular drivers in these tumor cells and in these tissues that are responsible for the tumorigenesis and for the tumor progression. Is this the case? If somebody else can confirm, can you hear me?
Yes, sir, we can hear you.
Yeah, Ziaul Hassan, could you please check your speaker settings? Thank you for the confirmation. So, as we already spoke about cancer is one area wherein precision medicine seems to be making significant headway. Right? New therapies seek to target the specific cellular pathway that is being exploited by cancer with a view to make something like a short life of tumor, or with a view to make intervention to the tumorigenesis and the progression of this cancer or tumor. So there exists an urgent need to identify new biomarkers or novel signatures that will help us in early detection and prognosis. New biomarkers and signatures can be and are in fact identified by leveraging publicly available omics data resources from the Cancer Genomics Association or TCGA, from NCBI resources, and from various other public resources, which you will get good introduction and good demonstration in this program to find the data and to download the data and to process and analyze the data so that we can find some novel associations with the disease progression. So the field behind this omics data analysis is the big data bioinformatics, and this has developed tremendously in the last several decades, that transformed life sciences and that enabled biotechnology, and also is empowering biomedical research to a completely new facet. So as a result, there came many new discoveries from researchers and from analysis of existing omics data. That includes new treatment, that includes diagnostic assays, that design strategies for novel experimentations that will confirm the effects of these identified biomarkers, treatments, and assays. So the main catalyst, as we discussed, for the transformation of the biomedical research is the emergence of sequencing technology. And in this program, we will introduce you to sequencing methods, and particularly next generation sequencing methods or high throughput sequencing technology that will help us study genomic variations, genes and their expression, RNA transcripts and their parts in the disease progression, and how all of these molecular elements are linked to disease biology. So to give you a sense of how this genomics data came into existence, then we already know that it's already been almost 50 years since the time of sequencing of the human genome. And in around 2015, the 1000 Genome Project brought significant number of whole genome sequences to the research community for analysis and for finding implications. That 100,000 whole genomes were sequenced by 2018, and today there exists, or there is an ever increasing number of data sets that are available to help researchers to understand cellular processes, to understand molecular functions, and in the context of disease biology, and in the context of, for example, rare diseases, and also in the context of cancer biology. So this growth of high throughput sequencing data is largely driven by the cheaper, faster, and more efficient sequencing of next generation sequencing, or simply NGS, right? From around $1 billion in 2001 to around approximately $3 million in 2008. And by 2017 alone, the cost of a single sequence genome was close to $1,000. And the aim is to reach around less than $500 or $100 by 2025, I believe, if I'm not wrong. So this has put a tremendous volume of data that is available, genomics data that is available, which is driving the whole big data bioinformatics field forward. So this technology like NGS can be used to explore the roles, the relationships, and the actions of various types of molecules that make up the cells of an organism. And many types of these omics data can be generated using, as we know, right, the next generation sequencing technology. And we will also study, or we will also discuss in this program, how this data can show detailed information about genomic variants and the patterns of gene and isoform expression and the differences in the microbial communities that drive the differences in the disease biology. So the economics of this data are hugely or extremely important for clinical practice and translational approaches of the implications that we can find and associations that we can find. So far, medical data as simply clinical data to introduce you to is a type of information that we have been using for centuries, right? It's like measuring temperature, measuring heart rate, measuring blood pressure, and other symptomatic data that is on the surface. These are typically easily and quickly measured or cheaply measured and are covered appropriately by insurance. But with the advent of molecular biology and cellular biology and with the next generation sequencing technology, we have learned how health and diseases are driven by what's happening at the subcellular level or what's happening at the intercellular level. And biomedical data refers to the combination of all of these omics data that characterizes the subcellular level of the disease biology with the clinical and relevant clinical data that is happening at the surface, right? And these different omics data include genomics, proteomics, and even microbiome of a patient that can be related to in the difference in which that these microbial communities help us process the metabolomics that we have in our body. So to give you an example of one of these omics data types and how they are used and how they actually change the approach. So as a method of measuring RNA translation, transcriptomics data or transcriptomics studies can actually help us detect mRNA expression inside a cell as an accurate alternative to measure membrane protein expression, which is actually used, for example, traditionally to, let's say, to characterize tumor histologically. This change in approach to focus on the molecular precision in a molecular level actually changed the precision in, improved precision in diagnostics, and it also enabled development of treatment based on molecular pathways instead of observational chemistry or observational characteristics of diseases, specifically tumor, for example. So the classification of tumor itself has changed, or it actually started to include molecular classification in addition to histological classification or functional classification that one can observe or that one can obtain superficially. And this includes, for example, classification at the level of molecules with complex variation across genomics and transcriptomics data, and also variation that will also include risk profiling of the patients based on the models that can be generated from the data trends that we find from these genomics, transcriptomics, proteomics, and even metabolomics data. And to give you an example, in addition to the existing PAM50 classification and histological classification of breast cancer, we now have a separate set of molecular classification for that characterizes breast cancer in terms of luminal A or luminal B, basal-like or normal-like or claudin-low, etc. So, and these are also described, or these are also demonstrated to be extremely important to classify, as several of these different classified subcategories react differently or respond differently to treatment options or therapeutic options. So the significance of the data that we just discussed is huge, as cancer can be diagnosed earlier and the treatment can be selected and adapted as the new data is generated, and the drug safety can be monitored, driven by the complex modeling of the disease progression themselves, not based on the superficial or not based on the what I'm looking for is response related data, but it's based on subcellular and system-wide data. Not based on the symptomatic data, but based on the subcellular, molecular, and system-wide data. That was what I was looking for.